Hello, everybody. Welcome to a new season of the Hellenic Initiative Against Alzheimer's Disease Bridge Seminars. Today's topic is about brain insulin resistance and the quest for specific biomarkers, while at the same time exploring the link between metabolic syndrome and dementia. I would like to thank Dr. Sotiropoulos and Professor Dallas for organizing and facilitating, as well as Genedis for sponsoring this as its 9th satellite event for Genedis 2026. We have very interesting presentations today, beginning by Dr. Aretouli, followed up by Dr. Kapogiannis. I would like to acknowledge my co-chair, Dr. Lyketsos, who will pick up the introduction after Dr. Aretouli's talk. So without further ado, let me introduce her. Eleni is an associate professor of clinical neuropsychology at the University of Ioannina. She has multiple research interests that focus on biological bases of psychological processes and health-related conditions on cognitive abilities on psychological processes. She has extensive research experience in Europe and the US, including a stint at Johns Hopkins, which was the most important stepping stone in her career, I say with editorialing. Eleni is an active member of several national and international scientific committees and societies that promote improved care for persons with neurocognitive disorders. Dr. Aretouli will be speaking about exploring the link between metabolic syndrome and dementia. Eleni, you're on and welcome. We can't hear you.
And just give me 1 minute to share my presentation, and that should be, I think it should be okay now. So good afternoon, everybody. Professor Lyketsos, thank you so much for the kind introduction. I'm really glad to participate in the Bridge seminar. Actually, this is my second participation, and I will be talking about the link between metabolic syndrome and dementia. I will make a brief overview of a few case studies, and then I will mention our own efforts to explore this link using the NAT dataset. And actually, these are very preliminary results that I hope you will find interesting. The metabolic syndrome is a clustering of commonly occurring disorders.
Can you put it full screen? Sorry. Full screen, please.
Yeah, we tried that, but I'm afraid I won't be able to see you. So, but do you prefer me to do so?
Can you do the slideshow?
Yeah, but I won't be able to see you on my screen.
It's just a little bit confusing right now.
Okay. I will do the full screen then. Is it better like that? And actually, is it okay? Okay.
Yes, much better.
And actually, I can also have a view of you, so it's fine. So basically, the metabolic syndrome is a cluster of commonly occurring disorders such as obesity, dyslipidemia, hypertension, and insulin resistance. And although this diagnosis has been questioned initially, it's been frequently used, especially in research. And there is 3 commonly widely used set of criteria for the diagnosis of metabolic syndrome from the WHO, the NCEP criteria, and the IDF criteria. Which all, despite their similarities, the main components of metabolic syndrome, they have some differences, and some say they end up creating different clinical entities. So, for example, in the IDF criteria, a very basic component is the central obesity, and then any 2 or more components are requested to fulfill criteria for metabolic syndrome, whereas in the NCEP criteria, any 3 components may be present and form and fulfill the diagnosis for metabolic syndrome. And then the WHO criteria have been considered not as practical because they actually require glucose intolerance test, whereas the other do not have this requirement. So there are differences between those sets of criteria. And so the question is, why should we investigate meds in relation to dementia and Alzheimer's disease? Its high prevalence is one reason. So 1 every 4 or 5 persons fulfills the criteria for metabolic syndrome. And then metabolic syndrome provides a framework to explore a potentially unifying pathophysiological basis for this cluster of risk factors. It is considered a practical and useful way to identify patients with multiple factors that place them at high risk of developing a range of diseases, including dementia and Alzheimer's disease. And actually, there is emerging evidence that indicates that metabolic disturbances have an impact on Alzheimer's disease neuropathology in various ways, both directly and indirectly. And finally, and most importantly, metabolic syndrome is associated to lifestyle, and so it is considered a potentially modifiable risk factor and therefore an important target for preventive strategies. In fact, the 2020 report of the by Livingstone includes those components that synthesize metabolic syndrome as important risk factors for dementia, and they suggest that if we were able to reduce those factors, we could potentially reduce neuropathological damage and thus prevents dementia. And what we know so far on the link between metabolic syndrome and dementia, so these are just a few key studies that were relatively recently published. So in 2019, a meta-analysis of longitudinal studies found no association between metabolic syndrome and all-cause dementia, but they did find an evidence of an association with vascular dementia in particular. But there was a certain important limitation in that. In those longitudinal studies, they had a relatively small follow-up duration of less than 10 years, which is considered a limited follow-up for this type of studies. And then in 2022, there was a study using the Whitehall cohort spanning a larger follow-up period over almost 30 years. And they did find that components of metabolic syndrome, but when present before the 60 years of age, are associated with increased risk of late onset dementia. They noted that the more components present, the higher the risk for dementia, but they weren't able to investigate whether specific subtypes of dementia were more highly correlated with the presence of metabolic syndrome. And then in 2024, the same association was explored using the UK Biobank data. The METS was defined by the NCEP criteria. That sample was very large. It was over 176,000 dementia-free participants, but they were older than 60 years at study entry. Their conclusion was that METS was associated with a 12% increased risk of all-cause dementia over a 12-year follow-up. Again, they noted that the presence of more METS components increased significantly the risk for dementia. And interestingly, they noted stronger associations between metabolic syndrome and dementia in those with low genetic risk for dementia, so ApoE4 allele non-carriers. And finally, a systematic review and meta-analysis in 2025 concluded that there is an increased risk of, for all-cause dementia and vascular dementia in particular. They didn't observe any association between metabolic syndrome and AD, but they did observe that specific components of metabolic syndrome, such as dyslipidemia, hypertension, and diabetes, were linked to Alzheimer's disease. And all previous studies and other studies conclude that further high-quality longitudinal cohort studies or case control studies are needed to clarify the association, this link between metabolic syndrome and dementia. So we sought to explore this link using the National Alzheimer's Coordinating Center data. This is, as you probably know, one of the largest, oldest, and most powerful Alzheimer's data sets. They have been collecting data for over 20 years now from 42 US ADRCs. And the data has been prospectively collected annually since 2005. So our first challenge was to create a valid estimate for metabolic syndrome, because there wasn't such a variable available in the NAC data set. And luckily we were able to compute such a variable using the IDF criteria. So a person had to fulfill the criteria for obesity and then meet at least 2 of the other conditions: high blood pressure, type 2 diabetes, or high cholesterol. And here you can see that I will not discuss further this slide. You can see the variables that we used to create this, the metabolic syndrome variable in the NAC data set. And just looking looking the distribution of metabolic syndrome by age in the NAC dataset, we note that the metabolic syndrome is less frequent in younger adults. So persons younger than 55 years of age have lower percentages of metabolic syndrome, whereas if we divide our sample at the 70 years of age, we see that percentages are almost, of those with metabolic syndrome, are almost equal. And the presence of metabolic syndrome by sex, we note that more men than women seem to have to fulfill criteria for a metabolic syndrome diagnosis in the NAC data set. So this 15%, almost 16 over here versus 13.5 for women. And then we saw the percentages of metabolic syndrome by ApoE genotype. And we note in this slide that those with 2 ApoE2 alleles have higher percentage of metabolic syndrome, especially if we compare them with those with 2 ApoE4 allele, which is a known genetic risk factor for Alzheimer's disease. And then we selected the metabolic syndrome characteristics at baseline, so for each participant at their study entry. And we looked at this data in relation to their cognitive status, but not cross-sectionally, but at their last available visit in the NAC dataset. So if a participant was followed up for, let's say, 8 years, we would consider his cognitive status at the eighth year visit in relation to their baseline metabolic syndrome characteristics at baseline. And so these are, I should say from this point on, that this is analysis and graphs contributed by the Bioinformatics and Human Electrophysiology Lab of the Ioannina University, and I'm very thankful for their contribution. So basically, in this graph, you can see absolute numbers. These are not percentages. But what we note is that if we follow the bars, the same colored bars, so for example, these are MCI persons, these are cognitively normal persons, these are persons with cognitive difficulties but not meeting MCI criteria, and this is persons with dementia. So interestingly, this group seems to have a higher percentage of metabolic syndrome compared to the normal controls or the other groups. Although these are absolute numbers, I think it's visually obvious. And then we selected only dementia-free participants at baseline. So basically we excluded everybody that was diagnosed with dementia at baseline. And again, the rationale was the same. We considered each person's characteristic at their study entry in relation to their last available visit, to their cognitive status at their last available visit in the NAC dataset. And so using only, the rest of the graphs referred only to dementia-free participants at study entry. And again, here you can see graphs on our metabolic syndrome characteristics and components. Actually, there were several variables. So there was clinician-reported diabetes, and there was self-reported diabetes. In both cases, and by cognitive status. So you can see the number of participants fulfilling metabolic syndrome diagnosis by cognitive status. And again, we note that there seems to be more MCI persons having diabetes than the other cognitive categories, diagnostic categories. Similarly, here you can see, again, in absolute numbers, how many persons in each cognitive diagnostic category have hypercholesterolemia. And again, we note that slightly more MCI participants seem to have hypercholesterolemia than the normal controls. And again, a similar pattern over here. This is the self-reported hypercholesterolemia at baseline in relation to their cognitive status at their last visit. And a very similar graphs for the hypertension, and again, a very similar pattern. We note again here that more MCI participants report having hypertension than normal controls. So these are persons who do not have hypertension. These are persons that do have hypertension, and these are persons that used to have hypertension. And this is self-reported, this is clinician assessed. And again, we note this higher presence of hypertension in participants with mild cognitive impairment. And here the use of antihypertensive medication seems to be also higher in MCI participants compared to normal controls. So these numbers refer to persons that do not use antihypertensive medication in comparison to those who do take antihypertensive medication. And a similar pattern for another metabolic syndrome component. Again, we note that the difference of the lipid-lowering medication in use in MCI participants is smaller than in normal controls. And finally, the body mass index distribution by cognitive status. So for the three groups, who doesn't seem to have very notable differences. Then we can see here an upset plot for the overlap between the three risk factors, the high cholesterol, diabetes, and hypertension. And what we note is that most persons in the NAC data set suffer from one or two conditions. So these bars over here show how many patients fall into its combination of those categories. So for example, here this number refers to the number of persons that have both cholesterol and hypertension, whereas these numbers, the left bars, show how many patients have a single condition. So this number refers to the persons that have high cholesterol. And so the triple overlap of risk factors is not as frequent, and the most common combination seems to be having high cholesterol and hypertension. And finally, here is an association of the baseline characteristics of the participants, the baseline characteristics that refer to metabolic syndrome with their cognitive status at their last visit. And so we note that it's a single component of the metabolic syndrome, and the metabolic syndrome itself over here seem to be related with the cognitive status of the participants at the last visit. This significant relationship remained even after correcting for false discovery rate due to multiple comparisons. And also, interestingly, the ApoE subtype was also related at the base. The ApoE subtype was also related to the cognitive status recorded at the last visit of each person in the NAC dataset. So these are very preliminary results. We are going to pursue further this analysis. We want to know what drives these associations, and also we want to know whether these associations are valid for dementia subtypes. So we still have further analysis to proceed with. And concluding my presentation, I would like to acknowledge the contribution of Professor Lyketsos, who helped me conceptualize this research project and formulate more specific hypotheses, and the Bioinformatics and Human Electrophysiology Lab, and Professor Exarchos in particular, who has been working closely with me on this project. And thank you all for your attention.
Thank you, Professor Aretouli, for a wonderful presentation. Let me point out to participants in the seminar that this is a great example of the value of the data core at Ioannina, and hopefully others will take advantage of it. I will turn things over now to Professor Alexopoulos. We're going to keep going and take questions at the end. So please write your questions down for Eleni.
Okay. So many thanks, Eleni, for this excellent presentation and for this overview, actually, of data pointing to the close relationship between the metabolic syndrome and dementia. The second speaker today is Professor Dimitrios Kapogiannis, who will talk about brain insulin resistance and the quest for specific biomarkers. Dimitrios earned his medical degree from the National and Kapodistrian University of Athens and received his residency training in neurology at the Massachusetts General and Brigham and Women's Hospitals, followed by a fellowship in cognitive and behavioral neurology at the National Institute of Neurological Disorders and Stroke. He is a senior investigator at the National Institute on Aging and Adjunct Associate Professor of Neurology at Johns Hopkins. His lab pioneered the isolation of extracellular vesicles enriched for neuronal origin from blood as a source of biomarkers for brain disorders. Dimitris, the floor is yours.
Thank you so much for this kind introduction, which I really appreciate this opportunity to share some of our research with you today. So I'll take a different route, and I'll go down to the molecular level. So we've been talking about insulin resistance. So to understand insulin resistance, we have to understand insulin signaling. So insulin signaling, it refers to a number of interrelated cascades, molecular cascades, that start by the binding of insulin to the insulin receptor. Immediately downstream of the insulin receptor is it's a key adapter molecule called insulin receptor substrate. IRS exists in two forms. IRS1 is the most common in the brain. Now, IRS1 has two functions. It can act as an adapter molecule that propagates signaling downstream, but also it can act as a blockade. And this is the main site of insulin resistance. So when tyrosine phosphorylations predominate in IRS1, then we have downstream insulin signaling. When serine phosphorylations predominate, there is a blockade, and this is the condition of insulin resistance. There is obviously a simplification. There are many other molecular factors, but that is the best way to approach it. Downstream of the IRS1, a number of cascades unfold. The AKT, GSK3 beta mTOR cascade. This is the so-called PI3K AKT cascade, or canonical insulin signaling. And then there is another MAP kinase cascade that has to do more with trophic actions of insulin. Now, there are, as everywhere in the human body, there are feedback loops. And so one very important feedback loop is that if you have overstimulation of this cascade, then there is a feedback loop through this molecule S6K that goes up and phosphorylates serines in IRS1, and so puts a brake. So too much insulin, the whole thing overruns, and a brake is applied. So you'll see this will become relevant as I go further. So what happens in Alzheimer's? Well, in Alzheimer's, there is a dysfunction of this system, a dysregulation of this system. Amyloid creates inflammation, local inflammation, neuroinflammation. Inflammation in the brain is manifest, among other things, by the presence of TNF-alpha, tumor necrosis factor alpha, which binds to some receptors in neurons and other brain cells and activates a different kinase called Junct. And so this Junct goes now and phosphorylates serine in IRS1 and implements a blockade to insulin signaling. So it's not insulin that does it, higher stimulation for insulin, but it's the presence of inflammation caused by a beta oligomers that creates insulin resistance in Alzheimer's disease. And this was documented, these pathways were better delineated in two publications that came in the Journal of Clinical Investigation more than a decade ago, which the authors there are showing a dramatic increase in markers of insulin resistance, especially IRS1 serine phosphotypes in MCI and even more in AD. Now, what does this all have to do with living humans? I mean, the previous was an autopsy study. Of course, if you have an autopsy study and you find these markers, then you can document the presence of insulin resistance. Well, the field has been trying to find biomarkers for insulin resistance for neuronal and brain-specific insulin resistance for some time. The first approach is to pretend and assume actually, I would say more pretend than assume that peripheral insulin resistance is what you can measure. And there have been several metrics of peripheral insulin resistance. The most common one is this called HOMA-IR, which is basically a calculation based on values of glucose and insulin in the blood. So that's easy. You take blood of someone, you measure their glucose, insulin, any lab can do it. Their ratio goes, not their ratio, I'm missing the word now. They both enter into this formula, and you have the metric of HOMA-IR, which is a surrogate of insulin resistance. So my lab and others over the years, they have used this HOMA-IR to look at parameters of brain function. So my former postdoc, for instance, Ariel Ouellet, in this study published in 2013, looked at HOMA-IR and its association with brain atrophy in Alzheimer's disease, and so found many clusters of atrophy in Alzheimer's disease. So the more HOMA-IR you had, the less brain volume you had in these areas. And here is an example, for instance, for an medial temporal lobe area. Further on with Oriel back in the day, in 2015, we looked at metabolism of glucose, FDG PET uptake, which is a surrogate of brain glucose metabolism and its association with HOMA-IR. And we found in Alzheimer's disease, the more insulin resistance you have peripherally expressed by HOMA-IR, the lower the brain utilization of glucose, the lower your FDG PET uptake. So is this, well, that was the best that we could do for a long time. Use peripheral insulin resistance as a metric of central insulin resistance and hope for the better. Now, I'll switch now gears, EVs, exosomes, and the others. So The cell produces a variety of vesicles as part of its normal physiological function and excretes these vesicles to the extracellular environment. So there are two main classes of vesicles, these called exosomes that are produced within a membranous organelle called multivesicular body, late endosome multivesicular body, that they are then released as exosomes, and then other vesicles that are secreted by budding of the plasma membrane. Now, these types of vesicles collectively referred to as EVs or extracellular vesicles. They basically have a common structure. They have a lipid bilayer membrane, some molecules embedded on their surface, and some cargo. Now, their cargo includes, interestingly, many signaling adapter molecules and signaling molecules from inside the cell. So, could that give us a window into the processes of insulin resistance, which happen through a propagation of signals in the cytoplasm? And the answer is yes. So, but before I go with that, let's refer a little bit to the methodology. So brain cells, like all cells in the body, secrete these EVs as part of their normal function in their extracellular environment. So if we see the brain parenchyma, there are all these cells, neurons, astrocytes, microglia, oligodendroglial cells. All of those cells secrete EVs in the intracellular environment, and some of them find their way into their peripheral circulation, into the blood. Now, we've been developing methods for isolating these brain-derived EVs. We start from plasma, implement different steps, importantly, immunoprecipitation. So we leverage specific antigens on the surface of these EVs that are of neuronal and other brain cell origin, and we're able to capture EVs that we think come from brain cells. So, what does this have with insulin signaling? Well, there are all these mediators, AKT, IRS1, GSK3 beta in the insulin cascade. More than a decade ago, we did several investigations to see what is present in EVs. And we found that in neuronal EVs, many of these cascade components are present, and actually they are present in abundance. So if we compare total EVs in blood, neuronal EVs, and epithelial EVs, we see that these all insulin signaling mediators are present, not only present, but a lot more of them than in other cells. Maybe it has to do something with the importance of insulin signaling for maintaining neuronal cells, but also the fact that these are cells that are very biologically, metabolically active. So, there are inside EVs, there are all these molecules that could potentially be surrogates for insulin resistance. So, also more than a decade ago, we did an initial study where we looked at the levels of phosphorylated IRS1 and total IRS1 in 80 patients and controls, and we found that total IRS1 was no different, but phosphorylated levels of IRS1, serine phosphorylated levels, were much higher, directly mirroring this neuropathologic finding. So that was the first study of its sort, looking at proposing IRS1 in EVs as a direct measure of neuronal specific insulin resistance. Then we conducted much larger studies. For instance, we leveraged the Baltimore longitudinal study of aging that has been going on for many decades. We identified subjects that at some point developed Alzheimer's in that, and went back in the freezers and took samples from when these subjects were still cognitively normal. We identified also some controls, and we pulled their samples, and we had a very large data set of 350 individuals, 128 of whom developed Alzheimer's disease at some point of their participation, and about double the number of controls. And we were able to go back in the preclinical stage of Alzheimer's disease by about 3.5 years. So what did we see there in this preclinical sample? We saw that the markers of insulin, dysregulated insulin signaling, IRS1, both serine and tyrosine phosphorylated IRS1 was higher in AD patients, in future AD patients compared to controls. These were from the time that all of these subjects were cognitively normal, but they're divided on whether they would go on down the road to develop Alzheimer's versus those that will remain controls. And we saw that this was true for across different ages of our participants. So also markers of IRS1 was negatively correlated with cognition, and this again mirrored findings from the neuropathologic study of Talbot in 2012. Their pathologic markers were negatively associated with cognitive performance. We saw the same thing with our EVs. So this is a busy slide. I'm just going to go fast. We also explored the relationship between these parameters and brain structure, and we found that these in 2 studies, this is a finding that we initially published in human brain mapping, and a few years later we validated for more markers. We found that these EV biomarkers were associated with brain volume. Also, something that it is important to note is that this is not specific only to Alzheimer's disease. We found that lower levels of this good IRS1, of this permissive insulin signaling mediator in PD, Parkinson's patients compared to controls. And very interestingly, this decrease was particularly true for PD patients that have cognitive impairment. So if you compare control subjects, PD with normal cognition, and Parkinson's patients with cognitive impairment, those with cognitive impairment had much lower levels of this permissive IRS1. So they had, in other words, defective insulin signaling propagation. And this was important for therapeutic reasons, because there was a clinical study conducted of exenatide, which is an insulin sensitizing drug that directly acts in these cascades and restores insulin sensitivity. And we were able to show that exenatide-treated patients in this clinical trial had a restoration of PY-IRS1, of the good IRS1, compared to placebo-treated individuals. And the same thing was true downstream in the cascade. Now, where does this leave us now? So there were some interim conclusions before I go further in more current research. So we found that EVs, neuronal EVs, contain insulin signaling mediators at various phosphorylation states, which may open a window into the brain and reflect insulin signaling in their parent neurons. These EV biomarkers suggest that insulin resistance underlie the pathogenesis of Alzheimer's, Parkinson's disease, other neurodegenerative diseases, as well as some psychiatric diseases. I won't have time to get into that. And that also these neuronal EV biomarkers can be used to demonstrate target engagement and predict treatment responses in a clinical study, such as the study that I shared with you. However, there were some unanswered questions. So, for instance, what is the relationship of peripheral versus neuronal insulin resistance, and how each one of those relate to factors of Alzheimer's pathogenesis? And is in neuronal insulin resistance, is this a causal abnormality, or is it just a non-specific finding, perhaps related to aging? So we sought to answer these questions in an ongoing study, and I'm delighted to share some preliminary results with you. So this is the Diabetes Prevention Program Outcome Study. It's a very large, long-term prospective observational study of individuals with pre-diabetes. There are something like 25 academic centers in the US are involved, and a few thousand of participants. So currently we're in the wave 4 of this DPPOS. which involves 19 years of follow-up. So people who had pre-diabetes 19 years ago when they enrolled, now they come back from the wave 4. Well, what happens in 19 years? These people were in late middle age. Some of them developed cognitive impairment, MCI or dementia, and many of them develop now type 2 diabetes. And so we leveraged this rich data set to have a project for the current wave of DPP-OS. And this is to measure markers of both neuronal and peripheral insulin resistance and look at their relationship to cognitive performance and cognitive diagnosis. This is an ongoing project. But we, before conducting the big project, we are conducting now a sub-project, which is to look at markers of neuronal and peripheral insulin resistance in relation to AD pathologic markers. And so for this sub-cohort, we have 456 participants, mostly women, fewer men, with a mean age of 73. And so we isolate two types of EVs in these participants, neuronal EVs and total EVs. Total EVs are a surrogate of EVs that come from every cell in the body, and we consider them a surrogate of what would happen in terms of insulin resistance in the periphery, everywhere in the body. Neuronal EVs are neuronal EVs. We think they're coming from the neuro. And then we isolate these EVs and we conduct two biomarker analyses. We measure p-tau, a beta 42 and total tau, and then we measure the insulin signaling markers. And so what is their relationship, if any? And so this is a very busy slide, but I will cut to the chase and try to digest it for you. To the left are insulin signaling mediators, are neuronal EVs and neuronal insulin signaling mediators. To the right are total insulin signaling mediators. And then we're looking in rows, the different Alzheimer's biomarkers, phospho-tau, total tau, a beta 42, a beta 40, their ratio. And we're looking at their relationship with neuronal and total insulin signaling mediators. So you see darker colors to the left and less dark colors to the right. So there you go, immediately something that catches the attention. We do see significant relationships for both neuronal and total insulin signaling mediators with these Alzheimer's markers. However, they're much stronger with neuronal EV. So the total insulin signaling markers can peripheral insulin signaling markers to some degree are associated, but of course this association is much tighter when it comes to insulin signaling markers within neuronal EVs. And some of these associations are breathtakingly, I would say, pardon my exaggeration, but I do find it actually breathtakingly strong. So for instance, if we're looking at serine IRS1 and phospho-tau 217, we see associations as strong as 0.78. These are Pearson correlations. So just to see it graphically, so for instance, here is p-serine IRS1, and here is 217 tau, and here is AKT, which is downstream. And so we see these very, very strong linear associations. So is this something to write home about? I would say yes, because if we look at, for instance, 217 tau to a beta 42, this is just about the same strength of association. So A beta and tau are related the same way that IRS1 and phospho-tau are related. So these are some interim answers. So I would say the question, what is the relationship of peripheral versus neuronal insulin resistance and AD pathogenesis? We found that neuronal EV biomarkers are strongly associated with EV biomarkers of amyloidosis and tau. Peripheral and neuronal EV biomarkers are moderately associated with each other, suggesting that brain and peripheral insulin resistance are dissociable. So if you have peripheral, we have some degree of insulin resistance in the brain, but there are definitely other processes that can drive insulin resistance in the brain. I think the most important of which is neuroinflammation. Sorry, let's go back. EV biomarkers of peripheral IR show low strength of association with EV biomarkers of amyloidosis and tau. And so is neuronal insulin resistance a causal abnormality or is it non-specific? Well, EV biomarkers of neuronal IR are as strongly associated with the presence of phospho-tau as a beta-42. For me, it's not proof. There's no causal relationship to be demonstrated by a biomarker study. But in my mind, these findings provide strong support to the hypothesis that there is a pathophysiological cross-link between AD and neuronal IR, which is actually a strong link, not a weak link. And with that, I would like to thank the hardworking members of my lab, some of whom are in attendance of today's seminar. It's their strong work that makes everything possible. And thank you so much for your attention.
Many thanks, Dimitris, for this excellent talk, shedding a light on the relationship between insulin resistance and AD markers or Alzheimer's disease, more generally speaking. So let's move to questions now. Are there any questions from the audience?
There are plenty, but
So,
I can start.
Yannis, would you like? Yeah, please go ahead.
I can start if you want. First of all, I would like to thank both for the really great presentations. And I think that's why I know why Costa suggests both of them to start after summer, because at least I gain at least 3 kilos, if not 4. If I'm trying to lie because probably they're 6, but anyhow. So it's a good reminder for us trying to keep our brain active, how bad things we can do over holidays in Greek islands or beaches. So, but seriously, I'm really scared. First, because Eleni, I'm really happy to see that we can in Greece use through the HIAAD, we use data. And I think that's a good example for others through the Ionian lab, the lab in Panagiotis Vlamus head, and with Themis, that we can do and work with this data. And that was very good, Eleni. Do you think that every of these parameters of metabolic syndrome are really related with the cognitive decline? Because Dimitrios, for example, said that, hold on, this is a neuronal signaling that happens and happens together. And that gives the overall question to all the clinicians about what we should care more when we measure, all these metabolic markers, which is more related to the peripheral insulin, about how we can help our brain, our cognitive reserve. So let's go to the specific questions. Eleni.
Okay, so first of all, I would like also to say how impressed I am by Dr. Kapogiannis' findings. And I don't know if I interpreted correctly, but it seemed to me that this might be a cognitive decline marker, regardless of the neurodegenerative disease diagnosis, which is fascinating. And so our approach, we don't really know what drives the association in our studies between metabolic syndrome and cognitive decline. I hope we will soon have more specific results. But it seems that insulin resistance, even if these peripheral measures, seem to be a strong contributor. We are going to assess with a lot of interest its individual component and how it relates to cognitive decline. We plan to also assess not only prediction of dementia, but also cognitive decline per se, like on neuropsychological measures. So I expect, from what I've known before and from what I've heard today, that even the peripheral measures we do have, that I expect there to be some kind of association and predictions that would be clinically meaningful.
Dimitri?
I want to thank Dr. Aretouli for this amazing presentation. It also scared me because I also have gained weight over the summer, and so. But I have to say, as a clinician now, to wear my other hat, interventions is one thing. Showing an association between a parameter, biochemical, biological, and cognition or the brain health is one thing, but whether you can actually reverse it by doing something is something that can only be answered by clinical trials. And so we desperately need clinical trials that will take any sort of interventions or many interventions combined, subject the population to them, and then see what happens. To some extent, it's happening, and it is the purpose of fingers and the pointer study and so forth. But the problem with such studies, which are the only way to really have proof that an intervention works in reversing cognition, improving cognition, and so forth, is the long follow-up. So you do something. How do we answer if you do something in your 50s, you're going to be okay in your 70s? Well, you need a 20-year-old study. And if you want to know in your 80s, you need a 30-year-old study. So that's the main difficulty to arrive in certainty for these things.
Last fast question, and then I will stop. Do you think, Dimitris, mechanistically that the correlation between the periphery and the brain could come from, let's say, exosomes coming from the periphery to the brain to dysregulate locally? Because there is a communication from the liver or whatever, of the fatty acid coming to the brain and regulate there, or you think it's a really neuronal deregulation that connects this tau or whatever amyloid beta AD-related mechanism, cellular mechanism, to the local insulin signaling is there?
I would say it's an excellent question to which I have no answer. It is, you and I can design like a whole 2 years of experiments based on this question. So
Okay, thank
remains
you.
to be seen.
Thank you.
Many thanks. Are there further questions?
Sorry, I see Dr. Skarmeas with raised hand.
Okay, Nikos?
Yeah, very fast because we're approaching the end. So really impressive talks. I think you somehow partially answered what I had in mind. So you think that it's really, the insulin and glucose dysregulation, you think it comes before the amyloid, the degenerative changes? Do you think it's a consequence? And I guess you already said that we need,
I think the best that I can say right now is that the process is. Oh, sorry.
Information from multiple times. If you can, do you think that that this regulation also defines a subgroup of people with cerebral amyloidosis with particular characteristics and maybe different responses, different prognosis, different responses to medication? So these are the two. They're more general questions, but yeah.
So thank you, Nico, for this question. So all we can say from these biomarker studies is association. So we can see very strong associations, quite honestly, much stronger than I expected, which tells me that the processes involved probably they're part of one cascade, not like a little detail out there that feeds there. So if I had to guess, is that and that's my leading hypothesis, but nothing more now, is that it's a beta insulin resistance tau phosphorylation. I think that it is insulin resistance may be a link between amyloid and tau. But that's the hypothesis. And then you can think of specific kinases, for instance, that like GSK3 beta, S6K, that are all known to phosphorylate tau. So if they get deregulated, they may phosphorylate tau and make it more prone to aggregation and so forth. So that's a hypothesis that we're going to pursue, and that's kind of an open question right now.
Many thanks.
Could I just make a comment?
Yes,
and then
yes, of course, Kostas.
So, so my comment, because Dimitri, you raised it about pointer and fingers. I think the fundamental flaw of both fingers and pointer is that they're not generalizable. They apply to people who are able to do the interventions, which, as we've discovered in ginger, is really tall order. So you're leaving out 95% of the population. You really don't know that the intervention would generalize to those people, which brings me to my suggestion to Eleni. I think you need to be doing prospective analyses. And I think hidden within the groups that you're studying are two very important groups. One are people who have no metabolic syndrome that do get dementia. So clearly you've got a pathway that does not involve it, and you might want to clarify to what extent is it genetic risk, et cetera. But you also have people with metabolic syndrome who do not get dementia. And so what is it that drives the resilience in these individuals? So I think those are going to be the kinds of interesting groups that you might want to focus on in prospective analyses. The NAC is now accruing blood samples. So in the NAC will be able to, as they go into their next renewal, to have access to blood samples for which tau and all those blood variables have been quantified. So as we send samples from Baltimore to the NAC, for example, they're going to send them to a repository that's going to run biomarkers, and then as well store samples so that you can pull samples and look at the relationship between the ATN biomarkers as well. It's 4 or 5 years away, Dimitri, before you can be doing EV studies. But keep in mind that NAC is becoming this gigantic, very important repository that we should all learn how to use.
Thank you, Kostan. We should do it together.
Yeah, maybe.
Are there any further questions? No, okay. So it is already 6:03 here in Greece. I would like to thank the two speakers for delivering these so interesting talks today. And many thanks to all participants for being here. See you in a month. Many thanks. Bye-bye.
Goodbye.
Bye, everybody.
Thank you.